back to second session of the second chapter, that is biological classification. In the first session, we talked about a system of classification, and we mainly focused on five kingdom classification, and we also discussed in detail kingdom Monera and kingdom Protista. In today's class, we shall discuss about the remaining kingdoms. So the instructional objective of this class is, so you'll be able to differentiate septate and aseptate mycelium, classify the kingdom fungi into 4 classes, explain alternation of generation in plants, list the distinguishing features of kingdom Animalia, reason out why viruses do not have place in Whitaker's 5 kingdom classification, distinguish viruses from virites, reason out why lichen are considered a pollution indicator. Okay, now the third kingdom, that is kingdom fungi. The characteristic features of kingdom fungi are these fungi are found everywhere. They are found in water, soil, air, and inside the animals and inside the plants. They usually grow in warm and humid places. That is why we keep our food in the refrigerator so as to prevent from spoilage by these fungi. And these fungi, the member of these fungi are filamentous with the exception of yeast, which is unicellular. And if you look into the body of the fungi here, there are number of slender thread-like structure called as hyphae, and network of this hyphae forms a structure called as mycelium. In the hyphae, you may observe a septate or septa or the cell wall or cross wall, septa or the cross wall. The hyphae having such a septa or cross wall are called as septate hyphae, and those hyphae which do not have cross wall, it will lead to the formation of multinucleate condition called as synocytic hyphae. They are aseptate hyphae or synocytic hyphae. The wall of this fungi cell are composed of chitin and polysaccharides. If you talk about the mode of nutrition, most of them are saprophytes or parasite, and they are also parasites, and some of them are symbiont, where the fungi live in association with that of algae to form a lichen, and some fungi form an association with the root of higher plant, and such an association is called mycorrhiza. The association is mutualistic, they benefit each other. Talking about the mode of reproduction, reproduction takes place by vegetative means, asexual means, and sexual means. And the spores produced in these fungi are present on the distinct structure called as fruiting bodies. Okay, so let us look at some of the asexual spores here. First is sporangiospores. Sporangiospores are non-motile spores which are produced in an enclosed structure called as sporangium. They do not have any flagella, so they are non-motile. And they are produced inside the sac, so called as endogenous. Next, conidia. These conidia are again non-motile structure, which do not have a common covering, so they are exogenous. They are non-motile. Third one is zoospore. Zoospores are produced in a structure called as zoosporangium. They are enclosed, so the production is called as endogenous. And when you look at each of the zoospore, they have 2 flagella, so they are biflagellate, so they are motile. And the other 2 forms of asexual spores are chlamydospores and oida. They both are non-motile. Looking at the sexual spores, some of the sexual spores in fungi are ascospores, basidiospores, acetospores, pseudospores, and teletospores, which are produced in different classes of fungi. And the sexual cycle in the fungi takes place in 3 steps. First is fusion of protoplasm between 2 gametes. The fusion of protoplasm will lead to formation of 2 nuclei in a single cell, like this. So this is the result of plasmogamy. After plasmogamy, 2 nuclei are there in a common cytoplasm, and this stage is called as dikaryon or heterokaryotic stage. And these 2 nuclei, the next step is these 2 nuclei will fuse together to form a zygote. That is fusion of 2 nuclei, we call it as karyogamy. This zygote being diploid undergoes meiosis to produce a haploid spore. Okay, so the third step is haploid. And these spores will germinate to produce hypha and mycelium, and then again the cycle continues. So these are the three steps in the sexual cycle of the hyphae. In some group of the fungi like that of basidiomycetes and ascomycetes, what happens is that after plasmogamy, karyogamy will not take place. Instead, it will keep on undergoing mitosis to form fruiting bodies, and later on in the fruiting body, they fuse to form zygote, and zygote undergoes meiosis. Okay, so such a stage, okay, where you have a very long period of dikaryotic stage or heterokaryotic stage is called as dikaryon phase. Okay, now let us look at the classification of fungi. Fungi are classified based on the structure of mycelium and that of the mode of reproduction. Fungi are broadly classified based on whether the mycelium has a cross wall or not. Those without any cross wall are called as aseptate, and this aseptate mycelium, okay, fungi are placed in a class called as phycomycetes, and this phycomycete is commonly called as algal fungi. Next, the septate mycelium. The mycelium with the septa are further classified based on their reproduction. If the sexual reproduction is known and if sexual reproduction is unknown. If the sexual reproduction is unknown in a particular group of fungi, they are placed in a class called as deuteromycetes or imperfect fungi. If the sexual reproduction is known, again they are further classified into classes like ascomycetes and basidiomycetes. So we shall talk about each one of them, that is phycomycetes, ascomycetes, basidiomycetes and deuteromycetes. First, phycomycetes. The common example is bread mold. You must have observed the bread mold on your bread. And these group of fungi are usually found in water. They are also found in decaying wood and moist area, damp area, and some of them are also obligate parasite. And looking at the mycelium, they are aseptate and cynocytic. Asexual reproduction takes place by zoospore formation or by ablanospore formation. Sexual reproduction also takes place by fusion of gametes. If two gametes are similar in structure, morphological similar, such a gamete are called as isogamous. And if two gametes are dissimilar morphology, then we call them as anisogamous or oogamous. So some examples are Mucor, Rhizobus and Albigo. Next is ascomycetes. Ascomycetes are commonly called as sac fungi. They are multicellular, but there is exception, that is yeast, which is unicellular. The mode of nutrition is heterotrophs, that is they are saprophytes, they are decomposers, they are parasites, or they are coprophilous. Coprophilous are the one which grows on dung. Mycelium in that case is branched and septate. Here in this picture you can find there it is septate and they are also branched. Okay, so some of the examples are Aspergillus, Claviceps and Neurospora and Morel. Morel, this is an edible mushroom here. Okay, ascomycetes. And talking about their reproduction, they reproduce by asexual spores called as conidia. And as discussed earlier, conidia are the non-motile spores produced exogenously. So here you can find here, these are the hyphae and group of this hyphae together. We call it as mycelium. On this, there is a formation of conidia. Okay, conidia are non-motile structure. Now, sexual reproduction takes place by formation of ascospores. Now, where are these ascospores produced? Okay, so we know that sexual reproduction involves the fusion of two gametes. Here in this, the first step of the sexual reproduction is plasmogamy. So two compatible mycelium, they come together and there is a fusion of protoplasm leading to formation of dikaryon. And this dikaryon, instead of undergoing karyogamy, it undergoes mitosis and produces a body called as ascocarp. And inside the ascocarp, there are so many SI like this. So in each SI, you can find two nuclei, okay, in a common cytoplasm here. And after that, when it matures, these two nuclei will fuse, that is karyogamy, and lead to the formation of zygote. Zygote will undergo meiosis. Okay, zygote is 2n. Zygote will undergo meiosis, results in the formation of 4 haploid spores, and each haploid spores undergo mitosis to give 8 spores. Okay, haploid spores, and these spores are the ascospore. Okay, so these ascospores are produced in the fruiting body called as ascocarp. Okay, next is basidiomycetes. Basidiomycetes, they grow in soil, on logs, tree stems, and in living plant bodies as parasite. For example, rust and smuts. The mycelium is branched and septate. Asexual spores are not found, but vegetative reproduction takes place by fragmentation. Some examples are Acaricus, Ustalago, and Puccinia. And looking at the reproduction in the basidiospores, basidiomycetes reproduce sexually by formation of basidiospore. And basidiospores are the basidiospores. These are produced inside the fruiting body of the basidiomycetes. Here, this is a mushroom here. Okay, they are produced here. And this fruiting body is called as basidiocarp. Inside this, you have so many gills here, and each gills we have number of basidia here. Okay, and in each basidia, again here there are 2 nuclei. Okay, that is because of the fusion of the protoplasm. So after the fusion of protoplasm, these 2 nuclei did not fuse, so it remained as a dikaryon here. Okay, so when it matures, these 2 will fuse to form zygote, and this zygote will undergo meiosis and give rise to 4 basidia. Again, they are produced exogenously outside. Okay, so in each fruiting body here, there are so many basidia, and each basidia produce 4 basidiospores. So these are the result of sexual reproduction here, and these basidiospores will germinate to produce mycelium, and this mycelium, again, if it is different strain, they both will fuse again here, that is plasmogamy. Plasmogamy is the fusion of the cytoplasm or protoplasm fusion, but the nuclei remain intact, they do not fuse, and that such a stage is called as dikaryotic stage, and it will germinate to produce mycelium, number of mycelium, and then they give rise to fruiting body. This fruiting body, when matures again, then only these 2 nuclei will fuse. So this is the steps in the sexual cycle of basidiomycetes, wherein they produce a sexual spore called as basidiospore. Now next is deuteromycetes, and they are called as imperfect fungi. Why do you think they are called as imperfect fungi? In the class of deuteromycetes, only the asexual spores are known, and that asexual spore is conidia. Sexual stage of these fungi are unknown. So that is why this group is called as deuteromycetes. Okay, that is imperfect fungi. And the myceliums are septate and branched. Once you know the sexual phase or sexual stage of these fungi, then they are removed from the class deuteromycetes and placed in either basidiomycetes or ascomycetes according to the sexual spores they produce. And talking about the mode of nutrition, they are heterotrophs. They are saprophytes, parasites, and decomposer. Some examples are Alternaria and Colicotrichum and Trichoderma. Next is kingdom Plantae. So here we'll be talking only brief part of the kingdom Plantae, because in the next chapter, we have whole chapter explaining about the kingdom Plantae in detail. So this kingdom Plantae includes all the eukaryotic organisms which are multicellular and which are photosynthetic. Okay, so they bear chlorophyll pigment, so that they can do photosynthesis. Now the life cycle in the kingdom Plantae, they are divided into haploid gametophyte and diploid sporophyte. What do you mean by haploid? What do you mean by diploid? So this is with regard to number of chromosomes. If there is one set of chromosome, then we call it as haploid. All the fungi are haploids. And if there are 2 sets of chromosome, which is usually denoted as 2n, then these are diploid. Okay, so diploid has 2 sets of chromosome. Haploids have 1 set of chromosome. Okay, so gametes, all the gametes are haploid. All the zygotes are diploid. Okay, now coming back to this plant kingdom, so they have a gametophytic phase which is haploid, and they also have sporophytic phase which is diploid, and they alternate each other. That is why we called it as alternation of generation. And one such example is given here with that of the life cycle of the pteridophyte. So in the life cycle of the pteridophyte, you can find that this plant body, okay, which usually we see here, this plant body produce a spore, okay, which are haploid, and this spore undergo mitosis to give rise to a haploid individual gametophyte, and this haploid individual will produce gamete, and gamete, two different gamete fuse together form zygote which is diploid, and diploid cell undergo mitosis to give rise to diploid individual. So we have a diploid individual sporophyte, diploid sporophyte producing haploid spores. Haploid spores later develop into gametophyte, and gametophyte, haploid gametophyte produce haploid gametes, and two different gametes fuse together to form diploid zygote, which in turn will give rise to diploid individual. So there is an alternation of generation here. Next kingdom is kingdom Animalia. So this includes all the eukaryotic organisms which are multicellular without cell wall, and they are usually heterotrophs. Okay, so all this diversity of organisms are placed under kingdom Animalia. Again, this detail we shall study in chapter 4. Okay, now let us just recapitulate this 5 kingdom classification of Whittaker and their characteristic features. So just by looking at this flow chart, you should be able to list out at least 2 features under each of the kingdom. Okay, so Monera include all the organisms which are unicellular and prokaryotes. Protista includes all the eukaryotic unicellular organism. Okay, and then talking about Plantae includes the eukaryotic multicellular with a cell wall, and they can do photosynthesis. And fungi are again eukaryotic multicellular organism which are heterotrophic which have a heterotrophic mode of nutrition. They cannot do photosynthesis. Okay, they are heterotrophs. And then Animalia includes all the eukaryotic multicellular organism without cell wall. Okay, so this is how Robert H. Whittaker has classified all the organisms into 5 kingdoms, and which is widely accepted. Now, we have heard about virus. So where are viruses placed? Where are viruses placed in the 5 kingdom classification? Do they have place? Okay, as you can see, it has no place in the 5 kingdom classification. Why do you think so? It is because viruses are non-cellular structure. These are subcellular structure, okay, which is made up of only nucleic acid and protein coat. Okay, so this nucleic acid acts as a genetic material, and the nucleic acid can be either RNA or DNA. Okay, and this virus, okay, also called as venom or poisonous fluid, and the name virus was given by Pasteur, and D.J. Ivanovsky, he was able to filter the virus through a bacteria-proof filter. And then another scientist called as M.W. Bijernik, in 1898, he was able to extract the fluid from the virus-infected plant, and that this fluid was able to cause infection in the healthy plant. Okay, so he called such a fluid as a contagium fluidium or infectious living fluid. And W.M. Stanley was able to crystallize the virus. So these viruses, having made up of only nucleic acid and the protein, once they are inside the host, once they are inside the living host, they are able to replicate. And once it is outside the living body, they are inert, they cannot replicate. So they form like that of a borderline between living and non-living. Okay, so they replicate only in the living host. That is why they are also called as obligate parasite. Obligate parasite. So if you look at the example of these viruses, here we have a tobacco mosaic virus, and this tobacco mosaic virus infects the tobacco leaf. Okay, so here the genetic material is RNA, single-stranded RNA, and then there are it is covered by protein called as capsids, and there are so many units of capsids here. Units in a capsid, each unit is called as capsomere. A number of capsomere together forms a capsid. Now next, another type of virus is bacteriophage. So bacteriophage are the viruses which infect bacteria. Here the genetic material is DNA. Again, there are protein coat. Now this one is, okay, as you all know, is causing pandemic. It's a coronavirus, and again here in this coronavirus you find, if you look at the structure, you find the genetic material which is RNA, and this RNA genetic material is covered by protein, and this protein, okay, it also forms an envelope, and this protein protect the genetic material, as well as it also helps in the, okay, infecting the host. It has a typical structure, that is a spike glycoprotein, and it forms a crown-like, so called as coronavirus. So the vaccination for this virus is not yet developed. That is why the world is suffering. And if you talk about the diseases in plants and animals, animal disease such as mumps, HIV, smallpox, herpes, and influenza. And in the case of plant, tobacco mosaic virus, chili mosaic virus, and then leaf curling and leaf rolling. So these are the diseases caused by viruses. These are, okay, disease caused. Now, other than virus, there is a viroid. Viroid is an infectious agent. It composed only of RNA, which is of low molecular weight, and the size is smaller than that of virus. It do not have protein coat. It has only the free RNA, which cause disease. And it was first discovered in 1971 by T.O. Diner. The example of disease caused by them is potato spindle tuber disease. Spindle tuber disease here. This is spindle shape here. Now, prion. Prions are a folded protein, again similar in size to virus. And these cause disease in cattle as well as human. So cattle disease is mad cow disease, and the same kind of disease is caused in human. We call the disease as CJD in human. Next, lichen. So this lichen is very special because it is composed of two different organisms, that is algal part and the fungi part. The algal part is called as phycobiont and the fungi part is called as mycobiont, and they live in symbiotic association with each other. They are inseparable. We cannot separate them. Okay, such a partnership benefit both of them. For example, like the algal part will do photosynthesis and give the nutrient to the fungi part. And fungi part, what they do is they absorb the nutrients and the moisture from the surrounding and pass it on to the algal part. So the symbiotic association between them is very special, and these usually grow in a clean area. If the area is polluted, they do not grow. That is why they are a very good pollution indicator. Okay, so that's all for today's class. So in overall, in the second chapter, we talked about, okay, let us just recapitulate what we have learned. That is, like we talked about the classification system, and then we just focused on 5 kingdom classification. And in 5 kingdom classification, the kingdoms are kingdom Monera, kingdom Protista, kingdom Fungi, kingdom Animalia, and kingdom Plantae. Okay, so all these kingdoms, out of that, we discussed about the first 3 in detail. And the kingdom Plantae and kingdom Animalia, okay, we shall study in detail in the next 2 chapters. Then we also talked about other groups which have no place in the biological classification. Okay, those are viruses, prions, and and viroids and lichen. Okay, that's all for today. Thank you.